because I'm going to pick up about where you left off, and I'm going to go into some of the details of the different gene drive mechanisms that have been pursued in mosquitoes over the past, I guess, 50 years or so, in the hopes of generating mosquitoes that either are no longer able to transmit pathogens that cause malaria or dengue, or gene drives that are able to effectively suppress the populations of those mosquitoes. And the different things that have been pursued have been, as Fred mentioned, driving an anti-pathogen gene into a population. This could be done into a neutral site where you would get successful drive would equal population conversion. So you would still have your mosquitoes in your area, but they would no longer be able to transmit malaria, or they would be no longer able to transmit dengue, and then disease transmission would stop. But ecologically, you wouldn't have much of a change. You can do the same thing without an anti-pathogen gene by driving your gene drive system through or disrupting a gene that's critical for pathogen transmission. So this would be a host factor that's required by a pathogen, so a receptor for a virus or another host factor that's important for the development of a parasite. And then successful drive would mean that that gene is no longer present in the host population, and the pathogens would die out as well. And again, ecologically, the vectors would stay in the population, but they would no longer be disease vectors. And then finally, you could use gene drive to disrupt a gene that's important for either both male and female development or existence, or just female development, and end up with only with males. And these are your population reduction or suppression strategies to try to reduce the breeding population and then hence reduce transmission. So, the CRISPR-Cas9 system that's been in the news quite frequently now is just one of many different gene drive methods that have been explored in dipterans, mosquitoes, and also Drosophila as a model organism for mosquitoes. And I've kind of categorized these into these cut repair type enzymes, which includes transposable elements, homing endonucleases, other designer nucleases like talons, and of course CRISPR. And then separate categories that don't rely on host repair processes for propagation, but I'm calling selective survival. So these are things like meiotic drive, X-shredder versions of homing endonucleases that don't replicate themselves but just destroy chromosomes, maternal effect dominant embryonic arrest, or MDIA, and then various underdominant approaches that Fred alluded to earlier. And just a little icon and what these have actually been successfully demonstrated in, mostly in flies, but a few through Austin Bird and Andrea Crisanti's group in Anopheles mosquitoes. So cut repair gene drives based on just what it says. So you generate a double-stranded break in a chromosome, and if that happens at the right time in the cell cycle, when a sister chromatid or a homologous chromosome is present as a repair template, then that can be copied and replaced. So in the case of transposable elements, an element can jump. If it's happening during DNA repair, then the sister chromatin can be used as a template that can restore the original gene, but you also can reinsert into a new spot on the same chromosome or onto a different chromosome, and the transposon will have increased in copy number. For a homing endonuclease or another designer nuclease, if you're able to engineer the gene in a homologous position from its target site, you can get double-stranded break generation and then copy and repair the same way. Cas9 is a special case because it can work in trans, because the nuclease activity is decoupled from the target recognition. So you can have a nuclease that's present in one spot in the genome, and then a guide RNA that's present in another spot in the genome. Cutting at that spot, you can get copying of the guide RNA along with any other cargo that's present, but not copying of the Cas9, which might already be fixed in the population or present at some level separately. Okay, one point to emphasize with all these cut repair-based gene drives is that in order to propagate themselves, they rely on accurate homology-dependent repair. And that is not the only type of repair that a cell can use to fix a double-stranded break. And this is going to be organism-specific. It's also going to be cell-type specific, developmental stage specific, as far as what type of repair is chosen to use. And this indicates how often you have a target site that's just disrupted by an indel type mutation, or whether or not you have completed repair and precise copying. And you can have imprecise repair, or you can have incomplete copying at some frequency. Now, if you're targeting a gene that's essential for the host, These kind of indel mutations will be strongly selected against and will disappear, so your target site will remain intact or you'll get copying. But if these are at a neutral location, these can accumulate and can serve to block the spread of gene drive over time. So the concept come about for using transposable elements based on the identification that the P element had recently invaded worldwide populations of Drosophila melanogaster. Oh, I see a hand raised. Does that mean I should be doing something?
No.
Okay. Okay. Researchers realized that since that had rapidly spread through fly populations, could this be done experimentally? Could you actually link a cargo gene to a P element and get that to spread into a cage population? And this is the first example of gene drive that was successful in a laboratory population. And as you can see, over the course of 10 generations and 4 different replica experiments, the number of P elements increased to almost 100% in those fly populations. And then they carried with them an alcohol dehydrogenase gene. So this was being spread into an ADH- fly population. And then that gene did not quite reach fixation, but almost, over the course of 10 generations. So this was a proof of principle that you could use a P element or a transposable element to drive a cargo gene into a population. And what they also found was that there were quite a few numbers of cases where the ADH gene was not copied correctly, or had been lost, or there was end joining repair. The P element had been moved around. And so limitations for transposable elements were quickly realized. Of course, you cannot control where it lands, so this can bounce around and end up disrupting other genes. The cargo size is limited, and eventually we'll select for P elements that are smaller to jump around. Defective copies can soon accumulate, and these can be silenced ultimately by host silencing pathways that handle transposable elements. And then when these types of experiments were performed in mosquitoes, elements like Hermes, MOS1, piggyback just did not remobilize well at all in Aedes mosquitoes, a dengue vector. Piggyback can be remobilized in Anofeles-Stevensii, the malaria vector, so the possibility of using that as a gene drive still exists. But again, because these cannot be controlled to any extent, I'm not sure that that's a favored method at this point. So a much more targeted approach to generating double-stranded breaks was pioneered in the use of homing-in nucleases. And here is a seminal paper using ISEE1. So by engineering homing-in nuclease target site into the Anofeles-Gambi chromosome and then putting the ISE1 nuclease on the opposite chromosome, and Kersanti and Burt and all were able to show that this was able to successfully invade cage populations of Anofeles-Gambi, but non-homologous end-joining mutants did occur at some low percentage, I think 1 or 2%. And so over the course of time, if this was done in a neutral site, again, those might accumulate. But the demonstration that this could occur in a mosquito vector is real. And so then just by re-engineering these homing-in nucleases to target endogenous genes that are critical, again, you could eliminate through selection any end-joining repair events, and then disrupt genes that are important in female fertility or female biology, limiting the fitness cost to only females. Because homing-in nucleases are more challenging to re-engineer, this group also explored the use of zinc finger nucleases or talon nucleases to drive through Drosophila populations. And what they found was that while homing did work and drive did work, end-joining events accumulated at a much higher rate, and that for the case of talons, they were not transmitted faithfully due to the repetitive nature of these talon nucleases. And so once copied, the rate of further homing was substantially lower, and these were not sufficiently good proxies for homing into nucleases. So to those extent, re-engineering homing into nucleases seems like the better bet at this point. But of course, that's when CRISPR arrived on the scene. So now your nuclease is just a Cas9 protein, and to re-engineer a new nuclease just requires a new short guide RNA specified here, and this could then serve as a new nuclease-based guide system. And of course, we've seen that this was highly effective, at least in a few generations in Drosophila through Ethan Byers' paper with scans. was just published in Science last year, and kind of started the ball rolling really on this whole system, as far as gaining a lot more attention outside the vector biology community. But the same kind of concept, a nuclease on one chromosome and then the target site on the other, and this was able to transmit itself to 97% or more of the fly population in just 1 or 2 generations. And so it remains to be seen how stable this will be in long-term fly populations, and then how this will move into other disease vectors to see if we can see the same kind of thing happening in anopheline or Adelman mosquitoes. So one of the issues that arises with any kind of cut-based repair system is off-target effects, and especially when we talk about CRISPR, where the guide RNA may have a lower specificity than a homing endonuclease, which is intrinsically coupled. Any off-target cutting can potentially result in allelic conversion at off-target sites, in addition to just lowering the fitness of the organism. So if you think about cutting in many places, then you can have chromosomal instability, lowered fitness. You can have the creation of novel inversions, novel translocations, simply by off-target cutting at sites that you didn't engineer. You can also have allelic conversion at off-target sites. So just like we had our scenario where Cas9 can work in trans to drive a guide RNA and cargo at a separate location in the genome, any off-target cutting where the homologous chromosome does not have the target site can result in copying of that site. So you could end up with off-target drive essentially happening at 1 or more sites throughout the genome that would be happening in parallel to the drive that you were designing. And without knowing what those off-target sites were, you wouldn't know what else you were driving into that population. So how do you identify off-target sites? Well, there's a lot of work being put into this in terms of human gene therapy, where this is a really important topic as well. And so while there are a number of programs available to do computational prediction of guide RNAs, the gold standard, I think, is going to be unbiased off-target detection, and this is one method that's been published recently by Keith Young's group, and where they take a double-stranded oligonucleotide, and when there is cutting, this can be incorporated into the genome. So wherever there is cutting in the genome, that can be incorporated. And then by isolating the DNA and shredding it, and then using standard Illumina-based library preparation, you can sequence specifically at where that oligonucleotide has been incorporated, and this is essentially an unbiased sampling of the genome. So when they did this, the results were fairly surprising. And I have one quote from the paper that was published earlier this year: the majority of identified sites were not detected by existing computational methods. And this is an incredibly difficult to read slide. I'll just zoom in on one part of it. You can see for a few different sgRNAs, the predicted site indicated by this black square had a few thousand reads, but then some of the off-target site predicted had a large number of reads as well, and then a number of sites that were not predicted also had the oligo incorporated. And over here you can see the on-target site actually was the minority. Many of these off-target sites and many sites that were not predicted at all were utilized or cut much more frequently than the on-target site. And as you can see from these different guide RNAs, some had a few off-target sites, some had many, some had barely any at all. And so evaluating what your off-target sites are in an unbiased manner prior to any strategy is going to be critical. Another thing to think about when we talk about off-target effects is actually genome size, because they're going to be related to each other. The larger the genome, the more likely you are to have more off-target sites for your guide RNAs. So when we think about the current data on the effectiveness of CRISPR-based gene drives, the data is coming from yeast, from Kevin Osveld's group, and from Drosophila from Ethan Byers' group. But we're looking at genome sizes on the order of 12 megabases to 130 megabases. As we start getting into disease vector mosquitoes like the anopheline mosquito, our genome size goes up, and goes up by a factor of 10 from Drosophila for Aedes mosquitoes, the dengue vector. And so the larger your genome, the more likely you are to have these kind of off-target sites, and the more this is going to become an issue. And as you start getting into other organisms like rodents or Asian carp or other organisms that have even larger genomes, again, knowing your off-target sites and reducing those is going to be critical. So the first step in any a repair-based gene drive is going to be this ability to insert your target sequence your Cas9 gene or your homing endonuclease gene into the site that you want it. And this is something that has been done with some regularity now in mosquitoes. It's been done both in Aedes mosquitoes by our group in this paper here, and by others either with zinc finger nucleases here or with CRISPR-Cas9 in these two papers, and it's been done in Anopheles as well. And so we do have the ability to land, basically, your gene drive cassettes in the right spot, whether it's a Y chromosome here or anywhere else in the genome, but we have not moved forward towards other types of gene drive. All right, so now I'm going to switch gears away from this cut repair-based gene drive and talk about some of the other gene drive systems that Fred alluded to that also have the potential to be used in other organisms but have been primarily pursued in mosquitoes or in flies as a proxy for mosquitoes. And one of these, and this is a paper that was described in 1966 on a naturally occurring sex disorder based on what we'll call meiotic drive. That is, when they looked at the number of males or females produced by parents of this, there was a strong bias towards male-specific progeny, and this could be inherited every generation. The males that came out of this also gave a strong male-biased progeny. And despite 50 years of effort, the molecular basis for this distortion remains to be identified. This could not be used in vector control ultimately because in the wild there were natural resistance alleles which were quickly selected for. But this kind of concept led to a lot of thought about how to create this kind of naturally, or how to create this artificially, I should say. And one way that's a similar type of output, but a much more elegant mechanism, is this X-shredding sex distortion that's been described. So in Anopheles gambiae, we have the fortuitous location of ribosomal DNA repeats on the X chromosome, but they're not present on the Y chromosome. If you're able to produce a homing endonuclease, in this case comes from a filamentous fungi, Physarum polycepharum, that actually has its target site in ribosomal DNA. So no modification was necessary other than destabilizing it so it wasn't transmitted to the embryo. But during the sperm development, you can now destroy X-based chromosomes using this homing endonuclease or any custom designer nuclease. And then when you have your sperm, you'll have Y-bearing sperm that are normal. X-bearing sperm that have been, the X chromosome has been destroyed, are non-viable. So if these sperm are used to fertilize an egg, nothing will hatch. And if these sperm are used to fertilize an egg, you'll get males. And this is actually quite successful by introducing these. And this is actually to a cage population. They were able to show that they were able to crash that population in short order. Okay, this concept, this special case where these ribosomal repeats are present on the X is kind of unique to these Anopheles mosquitoes. But this concept can be adapted for any X-specific sequence. So if you know a sequence, especially a repetitive sequence, that can be targeted on an X chromosome, then you can destroy that chromosome and yield further biased male progeny, which of course over time could suppress the population. Having repeats is better than having a unique sequence because as you have end joining repair accumulating, you'll still have enough target sites that you can destroy the chromosome, and so those end joining events will not be able to be propagated in the population. Whereas with a unique site, once you have end joining repair, then that becomes resistant to further cleavage. I want to add in another point here. So we have the first male determining factor was recently identified by my group as well as J2, and what this gives us is not only a unique landing site for a mosquito that does not have a Y chromosome, Aedes aegypti and other Aedes and culicine mosquitoes, like we mentioned, or Fred mentioned with the Culex mosquito that's transmitting malaria, is that these have homomorphic chromosomes, that is an otherwise autosome that has male determining locus somewhere on the chromosome. In this case, the locus is here on chromosome 1. But by identifying this male factor, we've actually identified a male-specific sequence. As you can see, in situ hybridization labels only one chromosome, not both. So this is a male-specific sequence that can be used to incorporate a drive that can be male-specific. And now we have the opportunity to exploit that locus or exploit this gene for male-only drive systems. We're able to show that overexpression of that male factor completely masculinized, otherwise normal females, turning them from, this is the abdomen of the female mosquito, and turning that into a male completely, and then seeing that in those females we're able to get complete development of testes, accessory glands, instead of ovaries. So moving on to other selective survival methods that have been successfully deployed, developed in flies in the laboratory, and this one called maternal effect dominant embryonic arrest. And so what this is a system based on a maternally derived toxin and a zygotic antidote that rescues that. And so the toxin in this case was a microRNA or a series of microRNAs that targets an essential gene, and silencing of that gene aborts embryonic development. The zygotic antidote is just the same gene again, but that resists that microRNA. And so now you can restore embryonic development. When these are together, and you can imagine this can be used to drive genes into a population if the cargo is presented in tight linkage to these. So what we found was that because this genotype does not survive, these can then persist, and this axis is essentially backwards. So this is a Medea-free allele, drops to 0 in these caged populations. Okay, and then, underdominance-based approaches. It's another form of selective survival, essentially. And this is, what a lot of people refer to as the original gene drive approach. This was Christopher Curtis, who hypothesized this back in 1968 and then paid for it in Nature. The idea being that the generation of translocations, where the translocation itself as a homozygous translocation is completely viable, whereas wild types are also completely viable, but where you have a wild type chromosome and a translocated chromosome, you'll have mistakes during meiosis where the chromosome segregation does not occur correctly, and so heterozygotes for those are completely unfit, whereas homozygotes for either genotype are completely fit, demonstrated here. This was attempted back in the 70s using radiation-induced translocations, but the problem was that these individuals were not fit enough. With the advent of more specific gene editing reagents, you can use CRISPR to generate potentially novel translocations where you wouldn't have the problem with collateral damage from radiation-induced translocations, suggesting you might be able to resurrect this strategy and generate translocations where you can use these for driving genes into a population. Or all the underdominance approaches have this nice spot where if you hit the sweet spot in between, you can actually destabilize the population completely by releasing homozygotes for your translocation, and as the population decreases, you just decrease the amount you release, and if the translocation starts becoming fixed, then you can start releasing wild types to get it back to this 50% line where the population is not stable and will eventually crash. Okay. Another form of underdominance, which kills embryos in a different way rather than missegregation during meiosis. This is now a bipartite toxin antidote system developed by Bruce Hayes' group. This was published by Omar Farag 2 years ago in Drosophila. But again, you have this maternal toxin, which are, again, a microRNA that knocks down a gene essential in embryonic development, and an antidote now located on the homologous chromosome, and then a second toxin, and then a second antidote. And when these are located together, you can create a system where when one of these copies is present alone, it's completely not fit because you have a toxin but not the corresponding antidote. And the same thing with the other gene, but when both are present together, then you restore embryonic development. So these, when both are present, you have wild type fitness. When only one is present, you have a substantial loss of fitness. And what they were able to show is that at release ratios above the threshold, you could get fixation. At release ratios below the threshold, then the gene is lost. And this is really nice from the point of view of what Fred was mentioning, that this is going to be something that when you're releasing within a city, at the boundaries of those release points, the frequency of this is going to be much lower, and it's not going to be able to invade nearby areas where you don't have those releases because the threshold will be below the level where it becomes fixed. So this will actually be disfavored at those edges, whereas where you're doing your releases, it can become fixed. Okay, this requires large release ratios. Okay, that's part of the deal, and so for some organisms, you will just not be able to do that. But for those where you can, and again, in terms of rearing large numbers of mosquitoes or flies, where that's possible to rear large numbers, you can then rear and fix this thing, or like I said, keep it around that midway point where the population is destabilized, but you don't have to worry about accidental release or this kind of uncontrollable release. And Bruce Hay, along with John Marshall, have put forth a few other theoretical scenarios. Here's your Medea. Here's your release threshold where it can become fixed. With the underdominance melanogastric, that threshold is a little bit higher. A few other where you take these toxin antidote-based systems and either make them sperm toxin or a female rescue, or a maternal toxin and a Y-based rescue, or X and Y, some of these other systems that have not been built yet, but have even higher release thresholds. Again, the higher release threshold is good because it stops accidental release from spreading, and it stops spreading across boundaries. So you have these site-specific gene drives, but they do require a higher threshold for initial releases. Okay, so what's the potential for each of these systems for remediation? That is, how do you remove this from an environment once you start releasing? Okay, for transposable elements, it's not currently possible. For nucleases like homonucleases or CRISPR-based gene drives, it's really not possible to remove them from a population, but if you're targeting, say, a female fertility gene, these might eliminate themselves as the population crashes. Maybe not to the point of extinction, because as Fred mentioned, extinction is probably not likely, but as you get to the point where the population is lower, you'll have stochastic events that might eliminate the gene from the population. Esvelt put forth the idea of reversal drives, but these are only for really repairing a damaged gene, so you can restore the gene that you disrupted, but these do not actually remove Cas9 nuclease from the population. Oops, sorry. For selective survival, some of the meiotic drive or extrader approaches, same thing. As the population crashes, these can be stochastically lost from the population. But once you get into things like Medea, translocations, and other underdominance-based toxin antidote systems, large-scale releases of wild type above the threshold will destabilize the population and then eventually drive your system out of the population, and you can return to a wild type state. But these do require a concerted effort, which requires resources and time to do. So summary, again, with the appearance of CRISPR, cut and repaired based gene drives are kind of in the news because they're very easy to generate and they require extraordinarily low numbers of releases, but we have no knowledge of their long-term behavior and including off-target effects and stability. So these still require quite a bit of investigation. Okay, re-engineering homonucleases, though difficult, is still possible. These, once they're developed, are much easier to manipulate and are likely to exhibit better stability and fewer off-target effects than CRISPR-based systems, although we'll see as that time moves on. But selective survival approaches to gene drive do not require homology-dependent repair, and thus are likely to be even more stable and more predictable in their behavior. And then underdominance-based systems are the least likely to spread upon accidental release and are also expected to be much, much more stable than these cut-repair-based systems. But these require the highest release ratios. And I'll just finish this with a nice graphic from a new chapter. This is from a book on genetic control in malaria that I've edited. This chapter comes from John Marshall and Omar Akbari, where they kind of plotted what these different parameters are. So the highest invasive, the least confinable, and the least stable are your transposable elements and your CRISPR-based gene drive systems. And oops, sorry, I have a very finicky keyboard. And then you have things that are highly stable and highly confinable towards this side. And a lot of these, again, have been pursued in Drosophila. Some have been moved into mosquitoes. But now we can think about how these might be applied to other systems, provided the genetics works out as Fred mentioned. So that's all I've got. I'm happy to take any questions. And again, I thank the committee for allowing me to participate in this workshop.
Thank you so much, Zach. That was an excellent presentation. So if members of the committee would like to ask Zach questions, now is the time to do so. We have a few minutes. Please remember to unmute yourself, or if you're having trouble unmuting yourself, go ahead and send me an email and we'll get you unmuted. Any questions for Zach?
Hi, Zach. This is Jason Delborn, and I have a question about the spatial limitations. I'm thinking about if a trait goes to fixation, let's say in a city, and the wild types are present outside that city, it seems that at the edge of the population where fixation has occurred, it's almost as if you've released a lot of mosquitoes into that environment. And so I guess I'm trying to understand why that edge wouldn't just expand outward at a very slow rate, why it would stay contained within any particular boundary.
Well, it depends on which system you're referring to. So with a CRISPR system or a homoendonuclease or transposon, it would spread through that edge. But with some of the other underdominance-based systems, their thresholds are so high that at those edges, you would probably have instability, and you would end up with population reduction because those heterozygotes would just not be viable.
Thank you.